Yeah. Right. Hey, hello everyone. Okay, good evening, good afternoon. So my name is Padmini Salgame, and I'm going to give you sort of an overview lecture on immunology. That's kind of a hard task, trying to summarize the discipline of immunology in like a couple of hours, but we'll try. So please stop me and ask questions. If you don't understand something, we don't have to wait till the very end. We have plenty of time. So let's get started. So why is immunology important? It's really this discipline that's really allowed us to make vaccines. And the most successful story is really the eradication of smallpox by vaccination. This pointer is not working. Excuse me? This pointer is not working. It's making a... Oh, sorry about that. I think it was on the other mode. So as I was saying, smallpox is a really successful, eradication of smallpox is a successful story. The vaccine really has allowed us to get rid of this disease. So what's the immune system? What are the cells that make up the immune system? So all immune cells rather arise from what's called a hematopoietic stem cell. This is a self-renewing cell that gives rise to two types of cells, the progenitor cells, the myeloid progenitor cells and the lymphoid progenitor cells. The myeloid progenitor cells then give rise to a number of different cell types, including your dendritic cells, the macrophages, which then differentiate from the monocytes. You have your neutrophils, eosinophils, basophils, platelets, erythrocytes. So all these are derived from these myeloid progenitor cells. On the other side, you have these lymphoid progenitor cells that give rise to your natural killer cells, the T cell progenitors and the B cell progenitors, which then give rise to your T helper cells and your B cells and another subclass of dendritic cells. So all these immune cells arise from a self-renewing hematopoietic stem cells. Okay. So what do these immune cells do? What is the immune response? How does it help protect an animal, the human? So it really protects us against infectious diseases, be it bacterial infections, it could be viruses, it could be fungal infections, or against the toxins secreted by certain bacteria. And it can protect us from, going from very small viruses to large parasites. So the different arms of the immune system, as we go along, we'll see how they help protect against these various, what we call pathogens. Okay. So when the host is sort of exposed to an initial infection, how does the immune system gear up? What are the various steps that occur? So we'll go through each of them. And initially, when the host recognizes a pathogen, the host tries to contain by using anatomic barriers. So to try and prevent the infection from taking place in the first place. When that fails, that infection occurs in the host, then this infectious agent is the host tries to remove it by use of recognition of preformed nonspecific effector molecules in the host. If that fails, then what we have what's called the early induced innate responses, which occur between 4 to sort of 96 hours. And what this involves is recruitment of various cell types to the site of infection. We'll actually go through each of this in more detail as we go along. So again, and that really, this early innate response helps to a great extent to remove this infectious organism or agent. And this again involves activation of what's called pattern recognition molecules on these pathogens, and activation of effector molecules, and initiation of an inflammatory response in the host. Very often this is sufficient to get rid of the pathogen, but very often you need, so up to here, this is called the early innate response or the innate response. But if all this fails, the host has what's called the adaptive immune response that then comes into play. So the adaptive immune response is very specific to a pathogen. The innate response, the same response would occur if you had, bacteria A or bacteria B, or, virus, or, it could be some amount of differences in the effector molecules, but they don't really specifically recognize various pathogens. But the uniqueness of the adaptive immune system is that it recognizes specifically a pathogen. So if you have made an adaptive immune response to pathogen A, the adaptive immune response will not recognize pathogen B. So it's very specific to a pathogen or an infecting organism. And so adaptive immunity has memory, and we'll come to that later, how that is then exploited in making vaccines. And the whole premise of vaccine is the ability of the host to make this adaptive immune response that can not only sort of attack the pathogen during infection, but later remember that as a memory response. And if there is another infection, make a much stronger and a faster response. Okay. So adaptive immunity occurs late because a few things have to occur in the immune system before those adaptive T cells and B cells are activated. And as I said earlier, they're very specific for the invading pathogen. And they also involve a step called clonal expansion. And that is what allows it to differentiate into memory T cells and clonally expand. So if you have one T cell that recognizes a pathogen, now after the first infection, you'll have many more recognizing that pathogen. So this clonal expansion and memory occurring during this adaptive response. So what are the physical barriers? So many barriers can prevent pathogens from crossing the epithelial layer and colonizing tissues. So we have the skin, gut, the lungs, eyes, nose, the oral cavity. So there's mechanical barriers, which include these epithelial cells that form tight junctions and do not allow migration of these pathogens. And there's the airflow, the movement of the cilia, say example, in lungs, tears. All these are mechanical barriers that prevent the pathogen from sort of attaching and establishing infection. There are also chemical barriers here, which include fatty acids, antimicrobial defensins, the low pH in the gut, pulmonary surfactant in the lungs, and various defensins present both in the lungs, the skin, and the gut, and in other oral cavities. Enzymes in tears and saliva also contain what's called lysozyme that can cleave bacteria and kill it. So these are mechanical barriers that include both mechanical and chemical that prevent the pathogen from establishing itself in the host. We won't go through a lot of details with this, but there's also microbiological. The normal microbiota can also be sort of a competitive barrier. So it allows establishment of pathogens in the host. Besides these barriers, there are also preformed molecules or cells that are there to recognize pathogens and get rid of these pathogens. And one of the most important cell type that does this is called the macrophage. These are, kind of eating cells. They eat up a lot of different things. So the macrophages, and the way they can do this is by expression of various receptors on their cell surface that allows them to take up or phagocytose these pathogens from the outside, take them in and get rid of them. So these are the various receptors that are present on the macrophage, including the mannose receptor, the complement receptor, lipid scavenger receptors, and dectin-1 that specifically can recognize fungal pathogens. So these are all receptors that recognize certain molecules present on the pathogens, so the pathogens can adhere to the macrophage. And when they adhere to the macrophage, they this is, for example, this is a bacteria here. It binds to these receptors on the macrophage. This then allows it to be phagocytosed into the macrophage. And following phagocytosis, it will combine or form what's called a phagolysosome. So it fuses with the lysosomal compartment inside the cell. This is the cytoplasm of the macrophage, and form what's called a phagolysosome. So the lysosome contains a lot of molecules that can kill or get rid of the pathogen. There are a lot of enzymes present in there, and this fusion also leads to activation of nitric oxide and superoxide in this compartment, and they are also antibacterial or can kill the pathogen. So by recognizing these pathogens that are present outside the cell, these macrophages can take in or phagocytose, eat the pathogen, and this phagosome that contains the pathogen then fuses with the lysosomal compartment, and this then allows for the bacteria to be broken down. You can imagine, these are some of the pathways then bacteria use to avoid. And so there are several pathogens that have learned how to avoid this fusion with the lysosomal compartment and then continue to live and replicate inside the macrophage. But these are the defense mechanisms that the macrophage can use to kill extracellular pathogens. Is that clear? Any questions? So as I was mentioning, these are the various antimicrobial mechanisms of the phagocytes acidification. So what the phagolysosomal compartment fusion does is that it brings down the pH of that compartment, and then now the fusion leads it to a bacteriostatic or bactericidal effect on the bacteria. Toxic oxygen-derived products are generated. Toxic nitrogen oxides are generated. There's antimicrobial peptides in this in the lysosome compartment. So once it fuses, it can act on the pathogen. There are other enzymes like lysozyme, and there are competitors like lactoferrin, which and vitamin binding proteins. These are molecules that will sequester iron. And for most bacteria, iron is the food they require to replicate. So by having lactoferrin, you take away its food. And this is all occurring inside the phagolysosomal compartment. Okay. So the macrophages are an important cell type that can phagocytose external bugs, bacteria, other pathogens. Another cell type that can do this very well are the neutrophils. So bacteria can also bind to neutrophils like the macrophages using very similar receptors on their cell surface. They can also phagocytose, and a similar process occurs where the bacteria can be killed within the phagolysosomal compartment. In addition to this process, the neutrophils also have another process to kill the bacteria called neutrophil extracellular traps. So as the neutrophil is phagocytosing and killing the bacteria, it itself actually can undergo cell death. But when the neutrophils undergo cell death, they release these extracellular what's called neutrophil extracellular traps that can actually bind and kill the bacteria. And it can do that by many different mechanisms. They release histones, which can induce pro-inflammatory signaling and sort of activate immune cells to kill the bacteria. And most importantly, they contain the antimicrobial peptides and such as defensins, catalycidins, which can then trap and kill extracellular bacteria. So the neutrophil is not only able to kill the bacteria intracellularly, that's within itself, but once it undergoes cell death, whatever is released can help act on extracellular bacteria and kill the extracellular bacteria. Okay, is that clear? You look confused. No. No. Okay. What's that?
Slides are just a little different. Slightly, the grade also was good. But,
The one that's posted today?
yeah.
It's the same thing. Did he post the wrong slide?
Also the same, but then there was some like.
No, this is not the latest slide. I sent the late. Did you check like
Just downloaded Canvas before 5 minutes.
I hope you didn't load the wrong one. Sorry. Is that okay? I can send the,
Yeah.
yeah, I will send. Okay, I've changed quite a few. Okay, let's see. That's maybe from last year it was posted at now. You have this now.
Yeah.
Um, so let's go back. So neutrophils use two different mechanisms, so they can kill intracellularly and bacteria that are extracellular, right? So another very important, so the macrophage, the neutrophils, these are all referred to as innate cells that come into play and try to get rid of the bacteria very early on following infection. Another cell type that also is referred to as innate cell is the natural killer cell. And the natural killer cells also have a very rapid response. They can also kill and detect and kill targets within hours or days of an infection. And they're slightly different from adaptive immune response because that takes, as I said earlier, much longer. And this may become clearer later, but they have germline encoded receptors, unlike the T cells and B cells, where there's rearrangement of their antigen recognition receptors. NK cells have germline encoded receptors. So these are pre-programmed. And they have non-specific targeting. They identify targets by a very interesting mechanism of missing self and recognizing stress signals. So one of their main targets is cells that are infected and tumor cells. Okay. So this is a quick summary, and I think you probably don't have the slide, but you can follow what I'm saying is, and then I'll send you the updated slides. So the mechanism of how these NK cells sort of recognize the antigen is normally there is this MHC class 1 on a healthy cell, and there are inhibitory receptors on NK cells, and they are unable to activate and kill the healthy cell. But in a tumor cell or cells that are sometimes virally infected, this MHC class 1 molecule is downregulated, and now this inhibitory receptor can no longer function as an inhibitory receptor, and the activating receptor is able to get activated and release these lytic granules, which can then kill the target cell. Was that? Okay. So NK cells, just remember, are important. They're innate cells, and they're important to kill or get rid of tumor cells and virally infected target cells or virally infected cells. Okay. So so you have these macrophages and neutrophils, which may be at the site of infection, trying to contain the infection. But sometimes this is not sufficient, and these infected macrophage and neutrophils need help from other immune cell types. And this process is called the recruitment of other effector cells, and this is an inflammatory response that's being initiated in the host. So the initiation of this inflammatory response, the phagocytic process, the process by which phagocytosis is occurring, is itself a trigger for the inflammatory response to occur. So you will appreciate that the immune response doesn't wait. It doesn't wait to see that, oh, can I clear the bacteria? Should I get on to the next step? It is trying to get rid of the bacteria, but initiating the steps that come subsequent steps. So as the phagocytosis is occurring, the macrophages are getting ready to recruit other cell types to the site of infection. So this process of phagocytosis itself is a trigger for the macrophages to become activated and to start secreting cytokines and chemokines that would allow for cells from the blood, immune cells that are traveling in the blood, that are present in the blood, to leave the bloodstream and migrate to the site of infection. And so now you have a lot of cell types coming out of the blood, lymphocytes, neutrophils, additional macrophages. They come here and then they try to help this infected macrophages from and help them to get rid of the bacteria. So as this phagocytosis is occurring, this inflammatory process has already been triggered and other cell types have occurred to the site of infection. Okay, and this process involves effector molecules that are secreted by this infected macrophages recognizing these bacteria or pathogens. Yeah. So how do these cells that are in the blood know that they have to come out of the blood to the specific site, the tissue, right? So what are the signals and what's the process that allows it to come out? So these cytokines and chemokines that are secreted from the macrophage is a signal for these endothelial cells that are close to the this region where where this infection is occurring to activate or to upregulate adhesion molecules. Oops, go back. So this then is a signal around the blood, the cells that are floating through this region then get the signal through these activated endothelial cells that, hey, we have to leave the bloodstream and provide some help to the infected cells here. So this process then is called migration or lymphocyte extravasation. Here, this is an example of lymphocyte, but all immune cells, macrophages, neutrophils, all use the same process or mechanism to get out of the blood. So there are four kind of distinct steps that are used. So this endothelium, right, when there's an infection, it will upregulate, as I said, some adhesion molecules. And because of this upregulation of adhesion molecules, that allows for these immune cells, in this case, the lymphocyte, to bind to the endothelial cell, okay? And this binding, so the cells that were moving in the blood, this binding now slows down its movement. But that binding is not very strong. So it binds, it falls off, and it binds again, and it falls off. So it slows the movement of the cells and gives it like a rolling sort of movement to these blood cells. So that's kind of the first step. Its movement in the blood is slowed down. And following that, the chemokines that are released at the site of inflammation, they activate these rolling lymphocytes. That's triggered by conformational changes that occur in other addition molecules here and change in what's called the integrin molecules on the immune cells. So what you need to remember is that there are effectors like chemokines that are secreted that then activate these rolling lymphocytes, okay? So they're bound here, but they're still not tightly bound. Now they become activated. And these activated lymphocytes now stop rolling and firmly attach to the endothelial cell, okay? So, and once they firmly attach, then they become arrested in their movement in the blood. So they were moving in the blood, then they slow down, they're rolling, and then they get firmly attached to an endothelial cell in the endothelial layer, right? And this step is mediated by high affinity binding receptors. That's all you remember. You don't need to remember the exact names, but it's high binding affinity receptors that allow for this step to occur. And once this arrest and adhesion has occurred, now there's what's called the transendothelial migration. So these cells can go between two endothelial cells. They can migrate into the tissue. Okay, so they squeeze through the endothelium and get into the tissues without really damaging the endothelial cells. So these are four kind of steps that occur sequentially that allows an immune cell that's in the bloodstream to go into the site of infection or into the tissues, okay? So as I was saying earlier, then it's clear that it's a macrophage that's infected. that releases the cytokines that then activate these endothelial cells that allow for leukocytes to extravasate to the site of infection, and then this often blood clotting in the microvessels, and then once the infection is taken care of, cells stop migrating out, the endothelial cells come back to their normal levels, the activation receptors that were activated are down-regulated. So together, these changes are what's characteristic of an inflammatory response, and which then allows the immune cells to act at the site of infection. Okay, and it's very often you see, heat, pain, and redness at a site of infection, and all this is this active inflammatory response going on at the site of infection. Okay. Any questions? Okay. So, how do these macrophages recognize these pathogens, right? How do they know that there's something there that, there are phagocytic receptors, so they will phagocytose, but how do they know that they have to become activated to make these chemokines and cytokines, these effector molecules? So this occurs through what's called recognition of pathogen-associated molecular patterns, pathogen-associated molecular patterns, or PAMs, which then leads to the activation of recognition of that by receptors on macrophages and immune cells, activate those immune cells. So these are the PAM receptor, the receptors on the macrophages that recognize the PAMs. So they are called toll-like receptors. And very interestingly, these toll-like receptors were actually discovered in the Drosophila. So Drosophila has a very similar system, the toll system that it uses to defend against pathogens, especially fungal pathogens. So that same toll receptor in the human toll-like receptor in mammalian system came much later, but originally it was discovered in the Drosophila. So there are many types of toll-like receptors. So these are innate receptors on the macrophage and other immune cells that can recognize PAMs or sort of repeat structures on the pathogens. For example, diacyl lipopeptides is recognized by the toll-like receptor 2 in combination with 6. In combination with 1, toll-like receptor 2 can recognize triacyl lipopeptides present on many gram-positive bacteria. Flagellin that is present in Salmonella, it can be recognized by TLR5. Lipopolysaccharide present on many gram-negative bacteria can be recognized by toll-like receptor 4. And there are also other toll-like receptors that are present in the endosome or inside the cell, which can recognize pathogens that sort of replicate within a cell, like viruses, which would be present inside a cell, replicate within the cytoplasm, would be recognized by these toll-like receptors. So you have a set that's expressed on the surface and a set of receptors that's expressed within or inside in the cytoplasm. And together they can recognize a large number of molecules present on various types of pathogens to trigger or activate macrophages and other innate immune cells. So this toll-like receptor is really sort of a link between the outside invading pathogen and the host cell activation pathway. Okay. What do these toll-like receptors do once they recognize their ligands on the pathogens? There is a whole process, activation process that occurs. And ultimately, what is important to remember is that ultimately the transcription factor NF-kappa B gets activated. It gets translocated into the nucleus and binds to, with other transcription factors, binds to promoters on various of various genes, cytokines, chemokines, et cetera, that then get activated, get transcribed, translated, and the protein is secreted from the cell. So this binding of the ligand to the receptor, here shown as the toll-like receptor 2, will activate NF-kappa B, which then leads to the activation or induction of inflammatory gene expression, various cytokines, chemokines, and an example here is example of an interferon, an antiviral interferon, which can then control viral infections. So these are innate receptors that can recognize various molecules and the pathogens, and they're not specific to any virus or pathogen. So many different viruses will have the same ligand that the toll-like receptor will see. Different bacteria, LPS is secreted by many different gram-negative bacteria that can all be recognized by TLR4. Okay, so there's no specificity, but there is some amount of diversity in the sense that there are many different toll-like receptors that can recognize different types of ligands. So the toll-like receptors are one of the most important innate receptors, but I just want to show this to emphasize that there are other innate receptors too listed here that can also contribute to this early innate recognition of pathogens. Okay. So this early innate response is occurring, inflammatory responses occurred, and we said following this is the activation of the adaptive immune response, right? So a key step in activation of the adaptive immune response is the transport of the antigen, that is molecules from those pathogens, things that the host is being attacked by, invaded by, those things need to be recognized. So those antigens from the pathogens have to be transferred to the draining lymphoid organs. And it's only in the draining lymphoid organs that the adaptive T cell and B cells will get activated, right? So how does that message, how is that carried? The message carried from the site of infection to the draining lymph nodes. Who does that job? How does that occur? And that transport, the key cell type to do that is the dendritic cells. So it's the dendritic cells that are present at the site of infection, take either the entire microbe itself or pieces of the microbe and transport it to the draining lymph node. And here in the draining lymph nodes, there are these naive T cells that can then recognize these dendritic cells carrying the message. And if there is a T cell that has found its, we'll see how that occurs. But if a T cell recognizes that there is an antigen that its receptor can recognize, then it will bind to and stay attached to the dendritic cell. It'll get activated, proliferate, and now you have a lot of T cells that are specific to this antigen that the dendritic cell has carried from the site of infection to the draining lymph node. Is that making sense? Yeah. So dendritic cell then is the bridge between the site of infection and the draining lymph node for activation of naive T cells. Okay. So so when I say activation of the adaptive cell, what do I mean? So adaptive immunity is made up of two compartments. You have the humoral immunity and the cell-mediated immunity. Cell-mediated immunity, as the name suggests, consists of cell types that are the effector molecules, like T helper cells and T cytotoxic cells. And humoral immunity mainly consists of B cells and the antibodies that the B cells make. Okay. So why are these T cells, and these T cells and B cells form the adaptive cells of the adaptive immune compartment. And as I said earlier, adaptive cell is very specific, right? If it's made against one pathogen, it'll always only respond to that pathogen and not to another pathogen. So what is it that gives it this unique specificity? And that comes from expression of specific receptors. So the antibody is really a Y-shaped molecule, and B cells are the cells that make these antibodies. and they consist of 2 heavy and 2 light chains, um, uh, 2 polypeptide chains, and these are linked by disulfide bonds. They possess 2 what's called the FAB region that actually recognizes the specific antigen, and an FC portion, which is called the constant region. So this variable region is very important. This is the region that binds antigens or these molecules from the pathogens, okay? So these chains give it that recognition ability for this antibody. And both the light chain and the heavy chain contribute to this variable region. And so this is it's called the V region, and the C or the constant region. And we'll see later that why do we even have a constant region in the antibody? What's the function of these constant regions? But for now, it's important to remember is that this variable region, both the light chain and the heavy chain, contribute to the recognition of the antigen coming from the pathogen or the microbe, right? And this variable region, there are regions within this variable region, and there's rearrangement that occurs, and that rearrangement of genes can give rise to a huge number of different B cell receptors. That what allows it to specifically bind to one or the other antigen. So this variability that's there in this region is what allows it to bind to different antigens, and there's thousands and millions of these different antibody types that can with different variable region that can recognize a specific antigen. Okay, so that gives it this unique ability to differentiate between different pathogens. The T cell receptor, like the B cell receptor, also has a variable and a constant region. The TCR is made up of an alpha chain and a beta chain. Here's your variable and here's your constant region. So again, both the alpha and beta chain contribute to the recognition of antigen by the T cell receptor. The T cell receptor is bound to the T cell, so it has a transmembrane region. Unlike antibodies, IgM can be bound to, and IgD can be bound to the surface, but most of the antibodies are secreted into circulation. Okay, but T cell receptor is bound; it's not secreted of the T cell receptor, so it has this transmembrane region. So both, you can see, have this variable region that gives the ability to recognize different antigens. So I keep saying antigens. So what are antigens? These are the molecules that are recognized by the immune response. So and within these antigens, there are epitopes which then actually bind to these to the receptors. So antigens can be complex macromolecules such as proteins shown here. So this is a large antigen, and it has these epitopes. So this epitope 1 here can bind to antibody 1, and this epitope here in red can bind to another antibody in red. And so large antigens will have many different epitopes that can bind to many different antibodies. Okay. So an antibody binds to the entire protein, whereas T cell receptor will not bind to the intact protein or the intact antigen. For the T cells, the antigen has to be broken down into peptides, and those peptides then will be presented on a molecule called major histocompatibility complex, or MHC, that then recognizes the TCR or the T cell receptor. So here, here you needed the intact antigen. If the antigen is degraded, B cells will not bind, because as you can see, the epitope is formed by different regions within this antigen. It's not contiguous, it's not linear, right? Whereas in the T cell, these are linear epitopes that arise from the broken down or degraded protein. And so that's a big difference between antigen recognition and B cells will recognize the antigen non-degraded. So whereas here they not only degraded, but they have to be in association with an MHC molecule, okay? Just to make it complex. So I talked about the FC portion of the antibody, right? So that FC portion can come in different flavors, and this is what's called the antibody isotypes. So these antibody isotypes, the FC portion is different, but the FAB, or the portion that recognizes the antigen, is identical, okay? So for an antigen A, you can have an IgG type antibody, IgE, IgA, IgM. So you can have all types of antibodies to that same antigen. And the reason we have different isotypes is each isotype can function, has different functions. For example, IgG can cross the placenta. IgE is more involved in allergic reactions. And in terms of protection, is important in protection against these large parasitic infections, since they can activate cell types that can get rid of these parasites. IgA is a dimer. IgA is usually found in secretions in mucosal sites. IgM is a pentamer and is very efficient in what's called a complement fixing, and can really activate cells in the presence of complement can be activated. So their functions vary, and important to remember that the antigen recognition doesn't change, but their functional aspect, the FC portion, is different when we say isotypes, antibody isotypes, okay? Another very important concept to remember is clonal expansion. So in an individual, if that person has not seen an antigen, then there are very few cells in that individual that would specifically recognize that pathogen or that antigen on the pathogen. So here you have your stem cells, gene rearrangement occurs, and now you have a number of different types of mature B cells, right? Each expressing a different antibody receptor and able to secrete different types of antibodies. So now say this host is infected by an antigen. This antigen is recognized by this particular receptor antibody on the B cell. Once this recognition occurs, this B cell now gets activated and it'll divide, and these cells will further divide, and this is clonal expansion. So you can see once this cell has been activated, it can clonally expand to many cells now. So all these cells are identical. They all have the same antibody that's being secreted or expressed on the surface, and they all recognize the same antigen. So now from one cell, you have clonally expanded into many cells, and some of them will go on to become memory cells. Some will become plasma cells that secrete antibodies and can survive for long periods of time. And so these are sort of antibody secreting factories that secrete a lot of antibodies. So this is an antigen-dependent process. The other B cells that are present here do not get activated. They will if they recognize the antigen somewhere down the road. But for now, it's only this antigen that was present in the host or recognized in the host. And so only this cell has expanded clonally, okay? So how do these antibodies that are made against antigens and pathogens really protect the host? So there are many different mechanisms that are used. For example, if a pathogen secretes toxins and you want to get rid of the toxins, so the way that's done is neutralization. So if a cell if this is a toxin that's being secreted, the antibodies will bind to the toxin, and that antibody toxin complex will be taken up by a macrophage, and the toxin will be degraded. So imagine a tetanus vaccine, right? So what would you have if you had if someone was immunized with tetanus for tetanus toxoid? You would have antibodies against the toxoid, which is very similar to the toxin, except that it's inactivated, so it's a toxoid. So that person who has received the vaccine has made a lot of antibodies to the toxin, right? So if that individual is now exposed to this toxin, is infected with, say, tetanus, the Clostridium tetani, the pathogen that secretes this toxin, that individual would have these antibodies in circulation, right? And that antibody can immediately bind to the toxin and get rid of the toxin. So that's how the vaccine has worked. It can neutralize the toxin. Is that making sense? So and then the antibodies can also bind to bacteria in extracellular space. So there's a bacteria here, for example, that cannot be phagocytosed by macrophages. Many bacteria coat themselves with molecules that then allow them that prevent them from being phagocytosed. So how do those bacteria? How can you kill those bacteria then, right? So here, where the antibody comes into play, and it's called the process of opsonization. The antibodies will bind to the antigens on the bacteria to which they were made, the antibodies. And then this complex, again, the antigen of the bacteria antibody complex will be phagocytosed and degraded within the macrophage. And also another way of sort of activation of antibodies or way the antibodies work is called complement activation. Bacteria can activate complement themselves, and so when complement is activated, it can sort of be deposited on the bacteria, and then this can be recognized. This antibody then binds to this bacteria with the complement, and then you can get phagocytosed through the complement receptor. So the antibodies have recognized the bacteria through the antigen present on the bacteria, but having the complement deposited on the bacteria now makes this complex more palatable to the macrophage. It can take it through the complement receptor. So here it was being phagocytosed by the Fc receptor, and here you're using the complement receptor on the macrophage to take up this complex and be phagocytosed. So there's both ways it's going inside as a bacteria antibody complex into the macrophage, but here it's the Fc receptor and here it's the complement receptor. Okay, and in both cases they get in the macrophage and then they're degraded inside the macrophage. Another mechanism that antibodies can provide help, particularly against tumor cells or against infected, say a virally infected target cell, is as I said earlier, the NK cells, right? But the NK cell functions can be enhanced through what's called ADCC, or antibody-dependent cellular cytotoxicity, where the CD16 on the NK cells can bind to the antibodies, and this antibody can now form a bridge and recognize through its FAB portion the antigens on the tumor cells, and this recognition then allows for activation of the NK cells and secretion of granules and killing of the tumor cell or a virally infected cell. Okay, so this process is called antibody-dependent cellular cytotoxicity. So NK cells can kill, without this, but in the presence of antibody, it's called ADCC, or antibody-dependent cellular cytotoxicity. Okay. So this is just a slide to sort of reiterate or emphasize how vaccines have been able to control a number of different diseases. In this case, just an example of diphtheria, polio, measles, all have been virtually eliminated, just not here, but in most parts of the world. And this is through successful vaccination. And successful vaccines work because of these memory B cells that you can make to the vaccine and antibodies. So Thomas, just go on. Let's move on. So we talked about B cells, how they recognize antigen, their receptor. The antibody is really the B cell receptor, antigen specificity, clonal expansion, and how do they actually work and provide protection in the host. So let's move on to antigen-presenting cells and T cells, whereas I told you earlier, T cells see linear peptides that have been broken down, and they need MHC molecules to present these peptides. So where do these MHC molecules come from? How does this breakdown of the peptides occur? How do the T cells actually recognize this complex of MHC and peptide? So the MHC molecules or major histocompatibility complex molecules you have two types, class 1 and class 2. Both of them have this groove where peptides of different lengths can sit inside this groove. Both these MHC 1 and 2 are tethered to a cell membrane. MHC class 1 is present on all nucleated cells, whereas class 2 is present on specialized antigen-presenting cells like macrophages, dendritic cells, and some B cells. Okay. These MHC molecules are highly polymorphic, and this, oops. So this peptide complex, right? Peptide MHC complex is what is recognized by the T cell receptor, right? But these MHC molecules are highly polymorphic. So all of us here have different combinations of MHC molecules. And so it's very important that the T cell development then requires an additional selection step. Because the T cell is seeing antigen MHC complex, that MHC molecule that it's recognizing in the host should be an MHC molecule that's present in that individual, right? So that T cell has to recognize a self-MHC molecule in association with the peptide. If it cannot recognize a MHC molecule and a peptide from another individual who's expressing a different type of MHC molecules. So for this whole process, so that it's really geared towards T cell receptor recognizing self-MHC and peptide, not just self-MHC, the T cells go through a process called positive selection and followed by negative selection to remove self antigens. And this is a very complex process, but to put it simply, there are two steps that occur. So here's your thymocytes, and these are the cells that will undergo. And I think this is you probably don't have this, but the figure that you have is very similar to this. So basically, what happens in positive selection is self-peptide and MHC molecules are removed. So in a host, you don't want the T cell receptor in the host to recognize a self-peptide and make a reaction, right? If you made a reaction to self-peptide, what would happen? What's that? Yeah, and what kind of disease would that be? Autoimmunity, right? So in autoimmunity, the host is recognizing self-antigens with self-MHC molecules. So you want to get rid of those cells. Of course, it fails in some individuals for various reasons, and you have autoimmune diseases. But what is going on in here is you're positively selecting here, and so anything, and then T cells that are, so the first positive has to occur before negative is you want cells to be selected for your specific MHC molecule, right? So that you have T cells that will recognize MHC, but you don't want an interaction with self-MHC and antigen, then you have autoimmunity. So it's a little tricky, and I hope you can understand the concept of positive and negative, right? So here, self-peptide by the MHC is positively selected. But so any MHC, but then here you negatively select, so anything that reacts with self-antigens are eliminated. So initially you want every TCR that can see an MHC peptide selected. But then you go to a second round, everything that's self-antigen, you want that eliminated with your MHC, okay? So you then end up with mostly T cells that will recognize self-MHC with a foreign peptide, but not a self-peptide, okay? And these cells then come into circulation, and this is what then will recognize your or get activated when there's an antigen from a pathogen being presented on an MHC molecule, okay? And there are two routes of antigen presentation for class 1 and class 2. And the reasons for why we have class 1 and why do we need a class 1 and a class 2 molecule, right? So class 2 molecules are involved in presenting antigens that are coming from the outside. So if you have an extracellular antigen, a bacteria that's coming from the outside, it's degraded in the phagolysosomal compartment right inside a macrophage. And that degradation generates a lot of peptides from that bacteria. And those peptides are then loaded onto class 2 molecules, and then they come to the cell surface, and then you have your self-MHC with that foreign peptide sitting in here, okay? So it's coming from the outside, is broken down in the cell in the phagolysosomal compartment, loaded onto the class 2 molecule, and then presented on the cell surface, okay? The MHC class 1, on the other hand, takes care of pathogens within the cell. It will get loaded, the class 1 gets loaded with peptides that are generated within the cytoplasm. So here, it's the proteasome that degrades the protein or the antigen. So it's the proteasomal compartment that will process the antigen, and those processed antigens then are loaded to class 1 and then come to the cell surface. So one case, it's the phagolysosomal compartment that breaks down the protein coming from outside, and proteins that are within the cell are broken down within the proteasomal compartment, or the proteasomes are involved in this. And then you get loaded to class 1, or in this case, class 2, okay? And you can see that you have taken care of both kinds of pathogens to be able to get loaded onto these antigen-presenting molecules, okay? So here now you have your MHC class 2, here's your antigen, which is then recognized by TCR, and they get help from other receptors. We don't need to go into the details, but basically you have your class 2 with its peptide and the TCR. So the TCR recognizes this complex. It will never recognize just the peptide or just the MHC molecule, okay? The TCR needs this complex. Similarly, the MHC1, TCRs that recognize the MHC1 and peptide is also this complex. There are special types of T cells that recognize MHC class 1 and peptide. Those are called the CD8 cells or cytotoxic cells. And these that recognize peptide with class 2 are CD4 T cells. They're mostly helper cells that secrete molecules. These are cells that can actually kill the target cells, okay? So you can see here a CD8 cell, its receptor can bind to this, in this case, MHC, say expressing a foreign peptide. In here, this could be a virally infected cell, a tumor cell. This T cell will then get activated, and when it gets activated, it will release a lot of molecules that will directly kill this target cell. Okay, and that's why also known as cytotoxic cells. Whereas CD4 cells, on the other hand, so here's your CD4 cell, your peptide antigen coming from pathogens that were degraded in the phagolysosomal compartment. This T cell will get activated, but it does not necessarily kill the target cell. Instead, what it does is it secretes molecules that will activate the cell, in this case, a macrophage, right? So this macrophage, for example, is infected with a pathogen, in this case, a mycobacterium, and the mycobacterium has not been killed. This macrophage still has a lot of mycobacteria. Let's look at this first. The infected macrophage here has a lot of mycobacterium. So now when this T cell is recognizing a peptide that has come from this mycobacteria being presented by class 2, this T cell gets activated, and the activation allows it to secrete this cytokine called interferon gamma. This interferon gamma will bind to a receptor on the macrophage and activate the macrophage. And when this macrophage is activated, it can now is able to degrade it. The activation process generates additional nitric oxide, superoxide molecules, and other effector molecules that can actually kill this intracellular pathogen. So if you look at a tissue, this is a tissue example where the T cells have still not been activated. There's no interferon gamma, and once the interferon gamma T cell activation and interferon gamma is secreted, you can see there's very little bacteria. This is a special staining that's used to detect these bacteria. You can see tons of them here, and in this case, you see very little bacteria. So clearly, this activated T cell is activating the macrophage to kill the bacteria. It's not killing its target cell, but activating it, unlike CD8 cells that actually kill the target cells. And that by killing the target cells, you have removed either the tumor cells or virally infected cells where the virus now can't replicate. Okay? So these activated T cells come in different flavors, and also, for the activation of these T cells, just this recognition of peptide plus MHC is not sufficient. It's the first step, important but not sufficient. Excuse me. So clearly, signal 1 is this recognition by the TCR of antigen and MHC class 2. And this is referred to as the antigen presenting cell because that's what's presenting the antigen. Then you need a signal 2, which is called co-stimulatory signal, which is a molecule called CD28 on T cell binding to CD80 and 86 on the antigen presenting cells. And these usually get upregulated following infection in antigen presenting cells. This signal 2 is important. And once the signal 2 is engaged, that's when these antigen presenting cells will secrete additional cytokines being secreted, and this forms what's called signal 3. So without this signal 1 having this interaction, or cytokines would not be made, and the T cells don't get activated, even if there's some cytokine in the milieu. This engaging this TCR is key because the signaling downstream is from the TCR. But you need the CD86 to prevent anergy or apoptosis of these T cells. And then you need the signal 3 to allow these T cells to differentiate into many different subsets or many different types of T helper cells. Okay? So without signal 1, having the other 2 is of no use in terms of activating the cell. What do these cytokines that are secreted do? The third signal allows this naive T cell to differentiate into many different cell types. Okay, and don't have to remember each of the names, but important to know that there are many subsets, different subsets that they get, different types rather, that they get differentiated into: Th1, Th2, Th17, T follicular helper cells. These are all helper cells, and a T regulatory cell. So now all these different types of T cells, the effector function is different. They make different cytokines. They act in different ways to help the host get rid of a pathogen. But remember, they all recognize the same antigen. So So like with the B cells, you had isotypes, right? Immunoglobulin isotypes, where all the isotypes are the same antigen, but the Fc receptors were different. And so the functions of those B cells were a little different, right? One could fix complement, one was important in parasitic allergies and protection against large pathogens. And some crossed the placenta, some were present in the mucosa. So similarly, in the T cells too, these are subsets of the same. So it's the same naive T cell, it has its TCR, and depending on what third signal it received, it becomes either a Th1, Th2, Th17, or T follicular, or a regulatory cell. So regulatory cells are also made because you don't want T cell activation to last too long, because inflammation is good, but then you want to control the inflammation. And so T regulatory cells are then generated to control these helper cells. Okay, does that make sense? Effector functions are different, but their recognition, their TCRs are similar. Okay, so they're seeing the same antigen being presented by the MHC molecule. Okay. And why do we need these different types of cells? As you saw earlier, having a cell that makes interferon gamma is useful if you have intracellular pathogen and you can activate the macrophage. If you have Th2 type cells, it's protective against helminths. These molecules, these cytokines that are made are protective against these large pathogens. Th17 is important in extracellular bacteria because they can recruit a lot of neutrophils to the site, which can then kill these extracellular bacteria. T follicular cells are important to help B cells to make antibodies, and Tregs, as I said earlier, really regulate or control all these other subsets. Oops. So how do these cytokines that are made by these various T cell subsets, how do they actually carry out their effector role, right? How do they signal? So cytokines signal through a pathway called the JAK-STAT signaling pathway. So many different cytokines, they all use, I mean, the receptors are different, the different downstream STATs and JAKs that are recruited to the receptor may be different, but overall it's the JAK-STAT signaling pathway. Since there's activation of JAK, downstream then STAT molecules are activated, which then go into translocate into the nucleus to activate specific genes. So what you need to remember is the cytokines can coordinate the immune responses and initiate downstream effector functions is through these cytokine receptors that, for example, here is a receptor that binds the cytokine, dimerizes, it gets phosphorylated, then it can recruit JAK, it gets phosphorylated, which then phosphorylates STAT. So it's a series of phosphorylation events that ultimately leads to translocation of STAT. So what you need to remember is JAK-STAT signaling important for cytokines signaling, and it's through a relay of phosphorylation events. And this cytokine, there's a receptor for interferon gamma, interferon beta. Each of the different cytokines has its own specific receptor to bind to. Okay. I'm just going to go through without a break. Is that okay? Good. Okay. I've alluded to memory immunity, memory cells several times now. So what is memory immunity? How is it generated? And what's the need for having memory immunity, right? So this is the course of a typical acute infection that's cleared, say, by an adaptive immune response, right? So for example, here is entry of the pathogen or a microorganism. It has established infection, and during the early phase of infection, the host is trying to control the infection. Innate immune response has been generated. There's a lot of innate immune response, and at this time, the adaptive immune response begins, or the adaptive immune response is activated. And once the adaptive immune response is activated, the bacterial burden or the level of the microorganism starts to go down. So antibodies are important to actually finally get rid of the pathogen, okay? But and then you go into a memory phase where some of the effector of T cells and B cells that have been activated, some of them will go into a memory phase or become memory cells. I want to point out, though, that without having an early innate response, you cannot have an adaptive immune response. So if an individual innate response is missing, this bacterial burden would be very, very high. And the initiation of the adaptive immune response either doesn't occur or is highly compromised or takes a much longer time to kick in. So innate response is very important. And part of that innate response is the chemokines, the cytokines that are triggered, that activate the macrophages, that activate the dendritic cells to migrate to the draining lymph nodes, right? So that's why the early innate is clearly important to induce your an efficient adaptive immune response. Yeah. So, for example, you're looking at antibody responses in an individual over time. So this is by measuring serum antibody, right? So say you have immunized this individual with antigen A, or this person has an infection with antigen A, right? So initially, if you look at the titer of the antibody, there's be very little antibody in the serum. But over time, you'll see that there's an increase in the antibody titer for this antigen, for antigen A, right? And then again, if you see in plasma, if you keep measuring over time, it slowly comes down and then it plateaus off over time. But around this time, if you immunize this individual, or if that person gets infected again with antigen, with a bug that has antigen A, and then you give this person antigen B, now you go back to the serum and start measuring the antibody titers now, not only for A, but for B in this individual's serum, right? What you see is the antibody response to antigen A, the titers go up very rapidly, and they are much higher than what you had seen here. So this is referred to as a primary response. So this is the first time that antigen is being presented to the immune system in this individual. But now if that person sees that antigen again, the response is very rapid and much higher. The titers are much higher than what you saw here. So this is a secondary immune response. But for antigen B, this is the first time the host is seeing the antigen, right? So this is a primary response, and so it takes much longer, and also the titers are much less than for antigen A. So this is not only immunological memory, but specificity, right? For antigen A, the host remembers and is able to mount a rapid and a higher response. And it's specific because this response is not going to increase the response to antigen B. So, and this is really the principle of a vaccine too, right? So if an individual has received vaccine A, they will be protected against that antigen, but not against vaccine. So you need to get a vaccine for antigen B2 or pathogen B2, okay? But the host will remember that and make a rapid response. In this case, say the antigen A was the vaccine, but here the antigen A was actually in the pathogen. So here you're getting the pathogen as your second exposure. Now you can make a rapid response and a higher response to clear the pathogen. Is that making sense? Okay. And also what's interesting with antibodies is that over time, they can, both the amount of antibody and the affinity to the antigen increases by what's called somatic hypermutation. So the binding of the antibody to the antigen gets better and better. So there's somatic hypermutation. And that helps the antibody binding to the antigen and the downstream effects much more efficient. But this sort of somatic hypomutation does not occur in the T cell receptor. Okay. Like with antibodies, T cells also there's increase in antibody the T cell numbers and activation of T cells in a memory response. So this is a primary response, say to Listeria to LLO specific or Listeria specific antigens, and you can see when there's a secondary response, there's activation of T cells and then there is the T cell numbers come down over time, and some of them will go in as memory T cells. So when there's activation of naive T cells, those T cells can become effector T cells or memory T cells. So this is a very simplified version. There's a lot of specific ways that these cells become memory cells. It's not this easy divided effector memory, but for sort of to understand the concept, basically memory cells and effector cells come from the same naive T cells. Some will go on to continue to do their effector activities and control the infection. And some of them will go into memory T cells. And again, memory T cells come in many different types of memory, subsets of memory T cells, and particularly difficult to study because in humans, these are these tissue-resident memory T cells that actually go and reside at the, say, example, there was a lung infection. And following the infection, memory T cells were made. A lot of memory T cells will be in circulation. But there is a subset of memory T cells that will actually go back to the lungs and reside in the lungs. So again, if there is a second exposure, then they can act very rapidly. So these are cells that reside at the site where the infection was initiated. And there's central memory cells, effector memory cells, and these tissue-resident memory T cells, all again recognizing the same antigen, but their effector functions are slightly different. There's also recently a memory subset called the stem cell-like memory subset, which has more of a proliferative nature. So they don't necessarily make a very rapid effector response, but they proliferate and are there for long-term memory compartment, right? So they kind of feed into the other memory compartments. They just, as the name suggests, they're very long-lived cells. So clearly then you have memory cells that come or are derived from the naive T cells are expressing the same T cell receptor here. So we have talked a lot about immune responses being beneficial against pathogens, right? But immune response can also be harmful depending on the nature of the antigen, right? So if it's an infection agent or if the immune response is being made against a tumor, clearly it's protective, right? So you have an infectious agent, you've made an immune response, it's protective. If you have deficiency in any of the compartments, you may, the individual can get recurrent infections. But there are certain individuals who will make an immune response, particularly the IgE type of immune response to innocuous substances like allergens. You don't need to make an immune response to pollen. Pollen's going to do nothing to you, but there are individuals who will mount a very strong IgE response to pollen. So these are responses that are not necessary and kind of harmful to the host. Grafted organ, right? So there's rejection. So organs are rejected because the host is recognizing the MHC molecules on the organ as foreign and rejecting it. Because as I said earlier, we all have different MHC molecules, right? And that's why the HLA typing and the MHC, you have to have a similar or matched organs. And then we talked about this earlier, if you make a response to self-antigen. And we talked about T cell positive negative selection, and there are many other ways that for both B cells and T cells, there are ways to prevent autoimmunity or these cells being activated to self-antigens, but obviously it fails in certain individuals, leading to recognition of self-antigens, leading to autoimmunity, right? And tumor we just talked about. So clearly immune responses are protective, but there are instances that they can be harmful to the host. I just want to touch upon this and now go into details. So besides systemic immune response, you also have mucosal immunity or responses in the mucosal tissues that we have. For example, the respiratory tract, in the intestines, the stomach, etc., where immune responses, mucosal immunity is important. And just to sort of highlight what's the difference between systemic and mucosal immune systems. We talked about systemic immunity, where you have healthy tissues, and for example, and if it's if a bacteria is introduced, for example, this skin here, if this surface wound introduces bacteria into the skin, we know there's going to be an inflammatory response is generated. Cells will come to the site of infection. Infection is terminated, and the tissue is repaired, and then it comes back to the healthy state, right? So normally nothing is going on, right? An immune response is initiated only if there is an infection and that needs to be cleared. On the other hand, mucosal tissues, the healthy tissue is protected by preformed molecules present in the mucosal tissues. For example, there's IgA that's present in these areas, and so local effector cells are present continuously at the site of mucosal tissue to prevent infection from being established, because mucosal tissue is an area that's constantly exposed to pathogens, right? So like the respiratory tract, you're constantly exposed to pathogens. So the mucosal system is ready to prevent and sort of get rid of the pathogens very rapidly. So as I said, the IgA molecules are there, preformed cell types are present in here. Of course, adaptive immune system is triggered, but the preformed molecules help to protect early responses. So they are adaptive immune cells, but they're functioning like the innate cells ready to go in the mucosal tissues, okay? And that's because these tissues are continuously or constantly exposed to pathogens from the outside. This is just a slide to put, I think I talked about this earlier. So immune cells are important kind of to clear bacteria, viral infections, etc. But there are cases when they can be harmful to the host. And I think we touched upon this earlier. So I think I'm going to, it's my last slide. Yep. Any questions, anything? Get to go home early. Okay, everybody's, suddenly I see a smile on everyone's face. Any questions? Yeah. Can you speak up again? Yeah. It's pathogen-associated molecular patterns. Pathogen, bacteria-associated molecular patterns. I can write it and send it to you or to the class. Yep. Pathogen-associated molecular patterns. Don't worry about it. It's basically what it means is these are molecules present on the pathogen that are recognized by the innate cell, the receptors on the innate cells. Yeah.
HC1. mentioned like positive selection. I was wondering do they have endogenous proteins that show with it when it's not in the
It's very complicated, right? So where are the foreign proteins coming from, right? It's so yeah, there's nothing in there. It's in the thymus. The foreign protein is not there, but there's a lot of. I mean, it's very complicated. But you ask a really good question. Basically, you want to the self antigens are there. That's easy, right? So what is it seeing that can then be recognized as MHC and foreign, right? So there are other sort of peptides, proteins in there that could give you that MHC foreign sort of recognition for the TCR. No, it's always something in there. Yeah, yeah. It's not the empty MHC. Yeah, yeah. Okay.